Hello, everybody. It's Mr. Cuddill. We're going to skip 8.3 as far as me lecturing on it. I think it's pretty straightforward. It touches on some of the concepts that we dealt with last time about exothermic and endothermic reactions and how the heat released, energy released from exothermic reactions powers endothermic reactions. And it throws in the specific example of ATP production, which is an energy source, that a temporary energy source that many organisms use, including humans, to hold on to that energy temporarily. You know, ATP is pretty cool. There's about 10 million ATP molecules per cell per second that are made. So much is being made all the time, holding energy inside those cells for them to do whatever they need to do, to move stuff around, to get rid of waste, to bring in resources, anything like that. You make about 180 pounds of ATP per day on average, which is maybe more than you actually weigh. So you can tell then that it doesn't stay there. It gets used and then recycled, and then it gets used and then recycled. And what I mean by that is that the energy in that final bond gets broken, harnessed, and then the ADP, adenosine diphosphate, plus an inorganic phosphate then get rebound together to make ATP again and again and again. In fact, I read once that ATP is recycled each ATP molecule is recycled about 340 times per day, which is pretty cool. So in the 10th edition of Campbell, I'm here on page 148. That's 8.3. Here it is. And then we're going to start 8.4. The meat of what I'm about to go over is 8.4. It goes for like 8 or 9 pages in the textbook, which is a lot in that textbook because it's super dense, heavy reading. Go slowly. Proteins are super important. We're going to see them for the rest of the course. We're going to do a lab on them. The whole intention is not to rush through proteins. If you were in class, I would threaten this, but of course I would never go through with it. I'm just trying to get people's attention because probably energy and momentum and enthusiasm are at an all-time low. I get it. I've been here before. This is just a picture that you're going to see in this section 8.4. How about I earn my paycheck and I'll go over it? How does that sound? Let's start with a song.
They're the protein catalysts in every organism and enzymes. Through enzymatic action, your metabolism is driven enzymes. Staphylococcus, jellyfish, tarantulas, and trees with vanilla activation energy. Enzymes, you and me now, enzymes. You got them in your cells when they do cellular digestion. Enzymes. You got them in your mouth and in your stomach and intestines. Enzymes. The thing in enzyme action upon is called the substrate. They fit like a human complementary shape. Enzymes, CF reaction rates. Enzymes. An enzyme finds its substrate at its active site. Enzymes. Held together in a column where they snuggle so tight. Enzymes. About to form a breakage to the active sites. Chemistry reactions become products into enzyme specialty. Product gets released, enzyme repeats its action readily. Enzymes. Like any molecule, it energy defines its function. Enzymes. Environmental changes shape to malfunction. Enzymes. Every enzyme has a pH where it catalyzes best. A pH equals enzyme activity direct. Enzyme also sensitive and easily observed. Enzymes. What you chill a certain point increases their efficiency. Enzymes. But too much natures that destroy their activity. Enzymes. That's why if you running has a dangerous situation, all that you can call it enzymatic conformation. Keep it 98% enzyme optimization. Enzymes. Enzymes allow break structure to glucose. Enzymes. If you lack the enzyme lactase, then you won't enjoy the lactose. Enzymes. It's acylglycemia and PKU disease caused by inherited enzyme deficiencies. Enzymes, they what everybody needs. Enzymes.
Couldn't you just listen to it all day? Friday's coming. And you can access this. Just play it as loud as you want in your car and scare all of your friends. I love enzymes. Enzymes are great. There's about 4,000 different enzymes that scientists have identified in organisms. They are the workhorses of life. They're proteins, and let's learn a little bit more about them. So obviously, there's going to be a substrate that goes into this little pocket here that we're going to call the active site, and the substrate is the reactant that's going to react, and then a product's going to be made in the end. So here's the substrate, here's the active site right there. It's kind of a little pocket. You can see here this blue figure is showing you all these amino acids all folded in a certain manner, very specific. The shape is super important. That substrate's going to fit in there just perfectly, or it won't fit in there at all. You can see then that there's an enzyme substrate complex. So the idea is that there's an enzyme. Added to the enzyme is a substrate. You get some sort of complex. And the reason why this is in the middle here is because you can see that things change. You can see maybe not that the red substrate's structure has changed, but you can certainly see that the enzyme's structure has changed, forming the enzyme substrate complex. This is called induced fit, and the idea is that that the key going into the lock or the substrate going into the active site actually induces a change of shape and a fitting of the enzyme. I'm going to press pause for a second and go over a few things. Normally what we see is the suffix "ase" on the end of a name designates that it's an enzyme. Now, often what that means is what we have here is the prefix is the substrate. So we could say red thing ACE. So this blue thing here is the red thing ACE. So it breaks down the red thing and it is an enzyme. This only occurs in reactions that were going to happen anyway, most often in exothermic reactions where we don't have to add energy in, but of course there are examples where that's different. But as far as enzymes in the most basic sense, just think of them acting on spontaneous reactions that didn't need any energy input. They would have happened anyway eventually. They're located in very specific places in the cell. We'll get to this in a moment. They don't change the delta G of reaction, and they do something called lower the activation energy. I think you know a lot about enzymes, but maybe not these last two things as they relate to these kind of thermodynamic graphs that we're about to look at. Also, understand that enzymes don't get consumed in reactions. They get recycled. In fact, there are some enzymes that work super fast. One that breaks down hydrogen peroxide in our bodies breaks down 6,000 molecules per second of hydrogen peroxide into water and oxygen gas. There's even an enzyme in the lungs called carbonic anhydrase, which I think binds water and carbon dioxide together and makes hydrogen ions in order to regulate the blood pH. But the point is that it does that reaction 600,000 times per second. I want you to wrap your head around that. You can imagine in your lung right now, this little thing going 600,000 times per second, doing its reaction, doing it the same way and the correct way every time, not getting changed or consumed. It's pretty amazing.
Enzymes enable molecules called substrates to undergo a chemical change to form new substances called products. Each enzyme acts on a specific molecule or set of molecules called substrates. Each substrate fits into an area of the enzyme called the active site. This fitting together is often compared to a lock and key mechanism. However, the enzyme changes shape a little to fit with the substrate. In the enzyme-substrate complex, the enzyme holds the substrate or substrates in a position where a reaction can occur easily. After reaction, the enzyme releases the products and can go on to carry out the same reaction again and again.
I know that's old, but it's good stuff here. The title of 8.4, it says, enzymes speed up metabolic reactions by lowering energy barriers. Just remember, what enzymes do is they speed up reactions. I'll show you what the energy barrier thing is in a second. So let's imagine that we have some sucrose, good old table sugar. You've seen it a lot so far this semester. And we pour it into water. We know that sucrose is made up, it's a disaccharide made up of glucose and fructose. We also know that if we added water into that bond between the glucose and fructose, we could do a hydrolysis reaction, adding a hydroxide group to I guess, to the glucose and a hydrogen ion to the sucrose, and we could split them. So, is that happening here? And the answer is no. This is an example of a reaction that's enzyme catalyzed. If we want to break down these sucrose or these like kind of natural sugar molecules, disaccharides. In fact, I read that this wouldn't happen for days. There's just not enough energy in the outside environment. If you kept this at room temperature and I guess normal pressures, it's just going to sit here and nothing's going to happen. But if we were to add the enzyme called sucrase, and you can see that in the picture on the slide, if we were to add sucrase, that would facilitate the breakage of sucrose into glucose and fructose. Now, how does it do this? Well, I told you the other day that what enzymes do is they're going to latch on to the substrate or the reactant, and they're going to stretch the bonds and make them easier to break. So it said earlier that enzymes lower energy barriers. So what enzymes do is they stretch or they orient this correctly or they do something to it so that there's less energy needed from the environment to break this bond than if it was just sitting like this. So in the case of sucrose, not enough energy in the environment to break that bond between the two saccharides or carbohydrates there in the disaccharide. But what sucrase does is it orients it correctly, it stretches this so that we need less energy to break it. So remember, it's not reactant and then the catalyst would be the enzyme creating a product. It's like this, where there's a transition state, and that transition state is going to be demonstrated right here by these squiggly lines. You see those squiggly lines up at the top? So the idea is that what the enzyme does is it stretches or orients the covalent bonds between the atoms in the molecule just so that they're more easily broken. And then you can see between the reactants and the products that the covalent bonding has changed. I wish I could point to this. So it was A bound to B, now it's A bound to C in the products. You see the bond forming way down there. Well, this is an example of a reaction. Maybe this reaction was catalyzed by an enzyme and maybe it wasn't. I'm going to tell you that the energy needed to break those bonds, shake them and break them eventually, is called the activation energy. That's what this is. That's what enzymes do, is whatever it appears. Enzymes lower the amount of energy needed to create that transition state, those wiggly, unstable covalent bonds, to break them to then get the product over there on the right-hand side. Well, this is shown in this picture. And so that picture we just saw and this picture that you see now are both in this chapter. And what you see with the red line there is that the energy of activation is actually lower with an enzyme than without an enzyme. And the idea is that the enzyme is going to do its thing and necessitate that there's less energy to, I didn't need much energy to break that covalent bond, less energy to break this bond than without it. If you get that, idea, that's kind of the thrust of all these two slides. Well, there's a couple other things that I want you to notice here. If you look way over to the bottom right, you can see that the delta G is unaffected by the enzyme. What that means is, remember what delta G is. It's what has happened with respect to free energy over the course of a reaction. So I'm going to turn this way. So if this reaction occurs over time, so let's imagine that it's time on the x-axis here going from left to right. We have the reactants, we have the products. Is this an exothermic or endothermic reaction? And the question is, do the products have more or less energy than the reactants? Well, it looks like here that if free energy is on the y-axis, it looks like the products have less energy than the reactants. So this is an example of maybe cellular respiration or exothermic reaction where energy is exited. What I want you to understand is that whether or not there is an enzyme, it's not going to change what happens with respect to free energy in this reaction, or it's not going to change the delta G. That's a super important point. So make sure you write down that point. Well, I'm going to press pause. Go ahead and you can go back a little bit if you need to. Make sure you understand these two graphs because you will see both of them on the test. So here's another example of one of these free energy progress of reaction graphs. I'll go ahead and draw this one. So quiet. Press pause if you need to. What's different about this graph than the one that we just saw? Well, you can see here that the products actually have more free energy than the reactants. So this would be an example of something that was endothermic, meaning energy went into this process or into the reactants as they proceeded towards products in this reaction. I get it that there was a transition state way up there at the top. Maybe there was an enzyme, maybe there wasn't. We can't tell. Looking at this picture, there certainly was a bit of energy called energy of activation energy that had to be put in there to break those bonds in the transition state and then reform what once was E covalently bound to F is now E covalently bound to G in the products. But overall, energy looks like it was added in. The products have more energy than the reactants. Example of this would be photosynthesis. Well, let me show you how this works. So I've told you a few times now, but I'm gonna display this to you. So how do enzymes actually lower this amount of energy needed to break these covalent bonds? Well, it can orient the substrate correctly so that often there are two things that come together, like in photosynthesis, water and carbon dioxide come together in the presence of light to make glucose and oxygen molecules. The point is that maybe there's another one right here, molecule, and then the atoms need to be right next to each other in order to kind of rebind covalently with the other atoms. And we've seen that in those past graphs. The point is that enzymes are going to orient this substrate correctly, so that happens with the most efficiency. You can see here, there's an enzyme that's surrounding, it looks like a nucleotide there. This one's a better picture. What the D and the R and the W's and stuff stand for here, those are coding abbreviations for different amino acids, and then the number is indicative of where they are on the long polypeptide chain. Obviously, you see here these kind of beta-pleated sheets, and it looks like amino acid 130 plays a role, amino acid 99 plays a role. You could look it up what W or R means with respect to which amino acid they are abbreviating. But the idea is that this is the active site of the enzyme, and those will create either hydrogen or ionic bonding temporarily with the substrate, orienting it correctly. It's very specific, I guess is what I'm trying to show you here. Remember, if the DNA was ever mutated, then following transcription and translation and the creation of this protein that we call an enzyme, then the amino acids would be a little bit off, the shape wouldn't be correct, they would have the wrong amino acid, and then you wouldn't get this beautiful active site ready to work on the substrate that's down there in the bottom right corner. Same thing here, you can see tyrosine in the 14th position, asparagine in the 99th and 38th position, holding this substrate correctly. One more example.
Lysozyme is a small enzyme that binds to polysaccharide chains and breaks them apart by hydrolysis. It has two structural domains. One domain is composed mostly of alpha helices, while the other domain is composed mostly of beta strands. The interface between the two domains forms a cleft in which the substrate binds. The structure shown here contains one of the products of the hydrolysis reaction. Lysozyme acts as a catalyst by adding a molecule of water to the bond between two sugars, breaking the bond. This reaction is catalyzed by two strategically positioned amino acid side chains in the enzyme's active site, glutamate 35 and aspartate 52. The highlighted group on the reaction product shown here would have formed the bond cleaved in the reaction.
All right, here's another way that the active site of an enzyme can lower the energy. I always want to say this backward. activation energy or energy of activation barrier. It can strain these bonds also. I've told you this before, let me show you an example of this. It could stretch the bonds, it could rock the bonds, it could scissor the bonds. Here's a kind of more specific example of the things that can happen to these covalent bonds in order to lower the amount of energy needed to break them. You can see that.
If oxygen and methane molecules are mixed together in the same container, they collide. But there is no reaction until a spark or flame is added. Why? To react, the molecules must collide with sufficient energy to break the oxygen-oxygen bonds and carbon-hydrogen bonds so new bonds can form. The additional energy needed to break existing bonds and start the reaction is called activation energy. An energy diagram can help show these energy changes. Initially, the oxygen and methane molecules collide but do not have sufficient energy to react. A flame can supply the activation energy needed to start the reaction. At this point, bonds break and new bonds form in an exothermic reaction that releases energy. The energy released supplies the activation energy for other molecules to react, and the mixture explodes. In the air, nitrogen and oxygen are continually colliding, but lack sufficient energy to react. Heat from lightning supplies the activation energy to break bonds, and new bonds form in an endothermic reaction that absorbs energy.
Enzymes are proteins which catalyze metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model. Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate. Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase catalyzes the following reaction between carbon dioxide and water. This reaction proceeds 10 million times faster with the enzyme catalyst than without. A water molecule is coordinated to a zinc atom in the protein. A proton is removed, leaving a hydroxide ion. Carbon dioxide binds to the hydroxide ion on the zinc, and the product is released. When a similar shaped molecule competes for the active site, it reduces substrate activity. This is called competitive inhibition. Adding more substrates increases activity.
We'll get to those concepts. We're going to do a lab on enzymes. Don't you love it when he says, rather like a spaceship docking into a space station? All right, this is a picture on, I think, page 173 or 174. I'm not going to go over it for you. I want you to press pause and talk to your favorite stuffed animal or your neighbor, if they're there, and go over this, please.
Hi, I'm Dr. Kiki Sanford, and today on Food Science, we're breaking down proteins. One of the most important processes in cooking is protein denaturation, or the breaking of a protein structure by the application of extreme conditions. Denaturation changes the structure of proteins and therefore the way that they react. Without denaturation, many delicious foods would never be possible. Proteins are necessary for our survival. We have to eat them to be able to create them in our own bodies. And to proteins, structure is everything. From their most basic to complex levels, structure determines the function of proteins. Many of the foods we eat contain proteins, which consist of amino acids. There are hundreds of amino acids found throughout nature, but only instructions for 20 are contained in our genetic code. The amino acids that organisms can biosynthesize differ. Some amino acids must be provided by diet. These are called essential amino acids. Proteins are created by connecting amino acids end to end, like the links in a chain. Then chemical interactions and bonds within the chain cause proteins to fold into knot-like formations. Proteins have 4 structural levels. The primary, chain of amino acids. Secondary is the formation of patterns within the protein. Tertiary are side chain interactions leading to the stability of single protein molecules. And quaternary is the association of multiple protein molecules or subunits. So in uncooked foods, proteins are normally all folded up. There are an almost infinite number of possible protein structures, but the primary structure determines the final folded shape that allows a protein to perform a particular task. Change the amino acid sequence even slightly, and it might affect the structure enough to create a new protein with an entirely different function.
There's only so much of Dr. Kiki that I can take. I'm going to tell you, I was in college for 9 years, and I loved every second of, I loved being a student. I loved wearing flip-flops, riding a beach cruiser, learning from the greatest minds in the world in their respective fields. It was hard my last couple years when I was a PhD student. I didn't end up getting my PhD. I got my master's degree in cardiopulmonary physiology. It's pretty relevant to this class. I don't even know if we'll go into the lungs and heart at all. I hope so. One of my professors, my investigators, pet peeves, the guy who I was working with for my PhD, was a title like this lady has on her lab coat. It says DR, and then it has her name, Dr. Kiki Sanford, comma, PhD. So the idea is that you should either have Dr. Kiki Sanford, or you should just have Kiki Sanford, PhD. It's redundant to say Dr. Kiki Sanford, PhD. Anyway, let's slow it down a little bit.
Hi, my name is Henry Gallagher, and this is a presentation of how enzyme is a living protein inside our bodies that helps us speed up reactions. It does this by lowering the activation energy of the reaction. This is milo. Milo is an enzyme. This is milo's activation site. This is where a substrate will bind on and cause a catalysis. However, only a specific type of molecule will fit this site. Oh, here comes one now. This is Ernest. When he sits on Milo, he will split into 2 separate molecules. And as you can see now, we have Pete and Bertrand, who are pushed every enzyme. Milo, as you can plainly see, is an enzyme designed to break apart substrates. Remember, Milo didn't start this chemical reaction. It would happen regardless of whether he was here or not. He just speeds up the reaction. Now, let's say we have another enzyme. Vary the enzyme. Vary will attract 2 separate substrates. Here they are, Sylvester and Orlando. Like Milo, Vary seeds the 2 substrates in his active site. However, unlike Milo, Vary fuses the 2 together. Now pushes 1 single molecule in. Steve.
Oh, this is more my speed. One of the greatest things about recording all these lectures in the summer is that I've been wearing sandals every time, and I don't have a mask on. Let's go, one more video.
Meet our chemist, Harriet. She has a chemical reaction that needs to occur more quickly. Our chemist has some processes at her disposal that can help her speed up her reaction, and she knows of 5 ways. And to remember them, she thinks back to her days as a high school student, and the day she got a date for the dance. Harriet was in high school studying between classes. She had lost track of time and was going to be late to class. Unbeknownst to her, Harold, who was just around the corner, was running late too. They both sprinted to class, and as it happened, sprinted directly into one another. Now, this was no small collision. They ran squarely into one another in such a way that he knocked the books right out of her hand. "I'm sorry," he said. "Let me help you with your books." He kindly helped her recollect her belongings and politely offered to walk to her class. You'll never guess who went together to the dance later that year. Yep, those two. So as we can see from this example, the key to getting a date for the dance is to collide with someone and knock the books out of their hands. Now you're probably already aware that not all collisions lead to dates for the dance, thankfully. The collisions must have two important characteristics. One, correct orientation that allows the books to be knocked from one's hands, and two, enough energy to knock the books out. Shortly after this incident, Harriet decided to tell me, her chemistry teacher, all about it. I noticed some interesting parallels between her story and chemical reaction rates, which happened to be what she was studying in the hallway the day of the collision. Together, we decided to set out on two missions. Harriet wanted to help all chemistry students and chemists remember how to speed up the rate of chemical reactions, and I, being the nice guy that I am, decided to make it my mission to help create educational environments in which more dropping collisions can take place to increase future chemists' chances of getting a date for the dance. In order to facilitate this improved dance date-getting process, I proposed 5 changes to all schools that parallel Harriet's 5 ways to increase chemical reaction rates. First, I proposed that we shrink the size of the hallways. This will make it more difficult to safely navigate the hallways and will cause more collisions than in larger hallways. And by increasing the number of collisions, we increase the likelihood that some of those collisions will have the correct alignment and enough energy to create a date for the dance. Now, chemically speaking, this is equivalent to lowering the volume of a reaction vessel or a reaction mixture. In doing so, the individual particles are closer together and more collisions will occur. More collisions means a greater likelihood that collisions with the appropriate energy and configuration will happen. Second, I propose increasing the overall population of the school. More students equals more collisions. By increasing the number of particles available for collision, we create an environment where more collisions take place. Third, we must reduce the time allowed between classes. Heck, let's just cut it in half. In doing so, students will need to move more quickly to get from one class to the next. This increase in velocity will help ensure collisions have the appropriate amount of energy necessary to ensure book dropping. This is analogous to increasing the temperature of the reaction mixture. Higher temperature means particles are moving faster. Faster moving particles means more energy and a greater likelihood of reaction causing collision. Fourth, students must stop traveling in packs. By traveling in packs, those students on the outside of the pack insulate those in the middle from undergoing any collisions. By splitting up, each student has some more area exposed that is available for a collision from a passive student. When particles travel in packs, the surface area is very small, and only the outside particles can collide. However, by breaking up the clumps into individual particles, the total surface area is increased, and each particle has an exposed surface that can react. Fifth, and finally, we hire a matchmaker. Is this colliding and book dropping too violent? Is there an easier way to get a date that requires less initial energy? And a matchmaker will help with this. The matchmaker makes it easier for a couple to get together by coordinating the match. Our matchmaker is like a catalyst. Chemical catalysts function by lowering the activation energy. In other words, by lowering the energy required to start a reaction. They do this by bringing two particles together and orienting them correctly in space so that the two can meet at the correct configuration and allow a reaction to take place. So to sum up, if a future chemist wants a date for the dance, he must collide with another person and knock the books out of their hands. And if a chemist wants to make a chemical reaction occur, the particles must collide in the correct orientation with an appropriate amount of energy. And both of these processes can be accelerated using the 5 methods I've described.
Thanks for bearing with me. We're almost done. Just understand, if we were to outcompete the substrate for the active site by putting some sort of an inhibitor in there, then we would slow down reactions. So there are such things called competitive inhibitors. They occur naturally. They also occur as drugs or poisons or things like this that are out in the environment. There's another way to disable the substrate from being able to go into the active site, and that's called non-competitive inhibition. And the idea is that shape is so important, specifically in the active site, that if we bind something to even a remote portion of the protein, and it binds to the amino acids and changes the shape of the whole protein, it will presumably change the shape of the active site, and the substrate won't fit in there any longer. So that's inhibiting the reaction from occurring, but it's called non-competitive inhibitors. We're not out-competing the substrate for the active site.
Terminal enzyme catalytic cycle is a term for enzymes in the active site, which defines for enzyme substrate.
The enzyme then facilitates the breakdown of the substrate to its products, which partner the enzyme, leaving the active site free to catalyze another substrate as the cycle begins again. Competitive inhibition occurs when the enzyme encounters a blocker, which mimics the properties of the substrate and binds to the enzyme's active site. Thus, when the substrate is encountered, the active site is not available for attachment and no reaction will occur. Non-competitive enzyme inhibition involves the binding of a blocker to the enzyme away from the active site. This binding causes a conformational change in the enzyme, altering the shape of the active site, which prevents the substrate from binding. No reaction will occur as long as a non-competitive blocker is bound to the enzyme.
Okay, we'll go over 8.5 real quickly. It goes quick. Understand that there's a lot of reactions that are occurring in every cell, and we need to be able to regulate them, and we have processes in place to do that. Either we regulate the production of the enzyme itself up front, or if the enzyme has been produced, we allosterically regulate it, which just means we regulate its ability to work really well by having a molecule maybe attached to it and stabilizing the great shape of the active site, or we can have another molecule attached to it and then change it and make it unable to do its job until we're ready for it to do its job, and then that little molecule will leave. And you'll see that picture in 8.5. This says chemical chaos would result if a cell's metabolic pathways were not tightly regulated. I totally agree. Here's a picture of just one metabolic reaction in humans that biochemists have described for us. Here's a corner of it. There's a lot going on here. Just be overwhelmed by all the complexity, and all the enzymes are facilitating these very specific reactions. The number one way that we regulate enzyme activity is by literally not allowing the enzyme to be made in the first place. And what this picture is trying to show you is that there are proteins that actually sit on genes, and they sit there, and they do not allow RNA polymerase, which is the thing that comes in and transcribes the DNA into an RNA message to then eventually do translation, make a protein. The point is that none of that happens because a protein is sitting on the DNA and it's not allowing it to occur. So we can switch genes on and off. We do all the time in all of our cells in order to make proteins that do stuff in the cells if we need to. Another way that we do this is through this whole feedback inhibition. You can see that the product O here is feeding back. Probably it's going to bind to the enzyme that facilitates the creation of M from substrate L. You can see that here, how they're all in sequences sometimes, and they're very complicated. This is a picture that I like to use the CliffsNotes book, so you'll see this in the CliffsNotes book. Just know this is feedback inhibition. It's also called negative inhibition. We are going to see this kind of in a little bit better picture. What we see here is that the end product, isoleucine, actually shuts off its own production by non-competitively binding to enzyme number 1. It looks like if you follow that red arrow to the left and up and then over, now enzyme 1 doesn't work anymore, and enzyme 2, 3, 4, and 5 aren't even needed, and intermediate A, B, C, and D aren't even made. And then eventually, probably that thing will wiggle off there, and now enzyme 1 will be free to facilitate this reaction. Well, let's pause. Go ahead and go over this stuff, please, in 8.5 and the last stuff that I went over, and we're done. Thanks for your attention.